The bacteria that live in our gut, also known as the gut microbiome, metabolize the food we eat and secrete molecules that can control how our immune system works. To better understand the effects of gut microbiome on people's health, we conducted a trial assessing the influence of beneficial bacteria on 200 healthcare professionals who experienced elevated levels of stress. Here is what we've been working on.
Within immediate consortium, we performed a proof of concept study because we wanted to test whether the administration of Akkermansia muciniphila strain MUC can affect the signaling within gut-brain axis in medical workers who are heavily exposed to stress.
For this proof of concept study, so we're going to be analyzing lots of samples from the patients and the volunteers, and we will also perform the integrative data analysis of the molecular profiles generated in the volunteers.
So our role was recruiting patients to the study, and after that we have to check inclusion and exclusion criteria, and we also have to get patient permission for join to the study, and we control whole process of the study during our package 3.
So we recruited the health worker, especially nurses, physicians, paramedics who works at hospital. They have the shifts, minimum 2 shifts on the month. It's really important that they have the stressful work, yes, and we want to check the level of the stress, and we check the influence of the supplementation of Akkermansia muciniphila and the relationship between when we give this postbiotic and how it's influence of the level of stress and their mental health.
They do have bad dietary habits. They have disrupted sleep-awake cycle, and these factors are influencing the signaling within gut-brain axis. So the disruption in the signaling might occur on the lower part of the axis, so they might experience functional gastrointestinal disorders, or on the opposite branch, let's say so they can experience emotional disorders.
So apart from taking the supplement itself, it was mostly the questions that were being asked about my diet, my lifestyle choices, so that's the biggest part. Also the body measurements that were taken quite regularly. Other than that, nothing really more, so it wasn't bothersome in any way. The biggest experience of mine is that I wasn't really feeling hunger for about 2 to 3 weeks, so the biggest issue was to remember that I have to eat in this time. Other than that, it was really a nice and pleasant experience since I've lost weight.
So what's exciting is actually moving from metabolic health or immune health to actually well-being. So that's a whole new shift in the type of applications that we're looking at for these bacteria, for these technologies. We talk a lot about precision medicine, but if we can actually show that not only this works, but it actually improves people's health and mental health, then you're shifting the lens towards health and not necessarily disease. You see what I mean? So we're more moving into preempting disease and intercepting disease, catching disease early on so that you don't even need to go and see the doctor.
My part in this project is mainly focused on isolation DNA from stool samples, and we are preparing from this DNA libraries for 16S sequencing, but also we're preparing libraries for further analysis like whole genome sequencing that will be performed in other place. We will be proceeding with ELISA tests, also from stool, so we will be detecting zonulin and cloud protectin. So we have all samples, and now we will go through the analysis to gather all the data to put them in the main base. In our part, the data are actually preparing from the scratch, so we need to isolate the DNA, and this part is very crucial because here will be some problem, then the data won't be complete. So we need to prepare those protocols perfectly to get the most precise information about the microbiome.
this is a huge milestone. I mean, having completed the recruitment and so on, so now we're ready to go. We're in the starting blocks to run the samples, to analyze the blood samples and the fecal samples from the volunteers. What does this mean? That means that we're going to finally be able to answer the questions we set out to ask when the clinical intervention was designed, and this is really exciting. It's a really exciting time because now we're going to be able to answer that question. I mean, can, friendly bacteria, can they actually be good for your stress levels and for your transition from health to disease? Can it be protective, or some of them, are they negative, are they risk factors? But here what we're testing is whether a particular bacterium, a next generation probiotic, is actually protective against stress.
I think that these data will influence in the healthcare because we will be able to generate more data from different analyses, from different labs. This will show us how the microbiome on different level works with our health.
Well, I feel quite great about it since, it's all about the patient and treatment of ill people, so that's great.
What is the challenge is the reflection, the questions we're asking, the strategies we want to deploy to actually answer those questions. So you start with a yes-no question, does the treatment work, does it not? But very quickly, once you have answered this first question, you daisy chain other questions which come up from a slightly different angle. Defining the proper analytical strategy, the data analysis strategy for that is usually the biggest challenge. It's digging deeper with the data once you've answered the first question.
They tackle the problem of diabetes, they tackle the problem also of obesity, which is increasingly more dangerous for our society in Europe as well as in the United States. So I think it could be a great addition to right now used conventional treatments.
So the significance is being able to zoom in and really have a sharp focus on particular cell types that would be responding to the probiotic, or to the enhanced precision at the cellular scale on the markers of stress, and also the markers of the response to the probiotic that we're testing, Akkermansia, muciniphila. And that, opens up a technical challenge. I mean, being able to process data at single cell resolution, I mean, that is a massive challenge. But I'm lucky to say that we have some of the best investigators in the field, so we should be able to crack this one.
Our goal is to provide comprehensive data and valuable insights into citizens' well-being and health status, allowing policymakers, healthcare professionals and individuals to make informed decisions about their health and how to better allocate limited resources. Visit our website and stay up to date on our latest discoveries on gut health.